And what we're going to do now is introduce the new system. And our new system is going to be the muscles. And whenever we start a new system, we always think about kind of functions. You know, what does the muscular system do for you? So, anybody?
You maintain your posture and
Yeah, posture for sure. I mean, right now, even if you're not moving and doing stuff, you're not slumped. Like if all your muscles turned off, you would totally just be like a bag of bones lying on the floor. And if you've ever had an animal, like if you've ever had a whole like a pet when they're being euthanized, the difference between an animal that's asleep versus an animal where the muscles are just turned off is very, very different. Even when you're asleep, there's some basic muscle tone where you can just feel the body has some tension in it. It's pulling the joints together. It's keeping you from slumping over. So posture. What else?
Produces movement.
Yeah, obviously movement. And movement in all sorts of ways. There's a skeletal muscle, which is moving your body around, but there's the movement of your stomach smushing up your food. There's the movement of, you know, the like things getting smushed along tubes. There is movement of your iris getting smaller or getting larger as it changes the size of your pupil. So there's all sorts of movement, both the voluntary we're used to, as well as a lot of involuntary smooth muscle going on in the body, squeezing the uterus to pop a baby out if you're giving birth or something. What else?
Circulation.
So if we're going to talk about the cardiovascular, you know, the cardiac muscle, we could say circulation, etc. You know, I would say in general, we're going to put that into the world of the cardiovascular system. But indeed, that is driven by the contraction of cardiac muscle. So it could go here as well. What's another one?
Breathing.
Breathing, yeah. I mean, breathing is going to be driven by the diaphragm pulling down to increase the volume and lower the pressure so air rushes in your thoracic cavity. It's going to be at the core there. In fact, we saw if your phrenic nerve, I mean, we did talk about the phrenic nerve, that one coming off the cervical spinal nerves. If those aren't sending a message to your diaphragm, you're going to die. C3 through 5 keep you alive. Other things, what if you get cold, what happens?
Shiver.
You shiver. So it's actually going to be part of temperature control. You know, muscle is just a motor or an engine. It burns fuel. In this case, the fuel is going to be ATP, and it generates movement. Just like your engine in your car, you pour in the gasoline and, you know, spin the wheels around and drive around. If you've taken physics, you'll know that any kind of engine is going to be inefficient. It will convert some of the fuel energy into movement, but then also a lot of that energy gets radiated out into the universe as heat. You know, that's why you've got a radiator in your car, and coolant running through there. You know, as your engine's running, yes, you are burning fuel to make movement, but you're also creating a lot of heat that's just you have to deal with. You know, muscles are the same. You know, at least half of the energy that's released by the ATP does not directly get converted into motion. Instead, it is released into the universe as heat. You've probably noticed that. Like if you're like cold outside, one of the things, normally you think of the muscles move and kind of a side thing that happens, part of the movement, they get warm because of this thing where, the conversion of fuel is not that efficient. But you can do muscles purely to make the heat. If you're shivering, you're not going anywhere. You're not moving to move. Your moving is to actually generate heat and take advantage of the heat. And you don't actually care that you're moving. In fact, it looks a little silly that you're shivering there. So temperature control, right? If you're outside and you just need to warm up, you just kind of dance around. You'll probably be a lot more comfortable. Hike around. So posture, movement, temperature control going to be at the core of driving your breathing and your circulation. So let's get into the details. We're going to talk about, one, like, how does muscle tissue actually generate contractile force? And we should mention, like, muscle only contracts. If you want to push, you do that because you have muscles connected to bones. Like if this is your arm and here's your hand, if I want to, actually, that's a crappy drawing. If I want to like push out, I can't make a muscle actually push. Instead, what I do is I have a muscle like, the triceps brachii on the outside of the elbow joint, and this muscle contracts. But as this muscle gets shorter, it actually is going to extend the elbow and make it look like it's extending, it's pushing the hand further out. But the muscle itself can't ever push. Muscle only contracts. It also means muscles always have to be connected up in what we call antagonistic pairs. So if you want to have your arm be able to either bend your elbow or extend your elbow, you need to have a muscle that's on one side of the joint to pull, to flex your elbow, bring it up. You need to have another antagonistic muscle that works on the other side of the joint. So when that contracts, it'll extend or unfold the elbow. So because muscle only contracts, it's essential, that it's connected up into our skeletal system to create all the different elaborate movement that we can actually do. And particularly given our situation, normally I spend a little more time talking about this, but I think we're gonna go more into the physiology of how does muscle tissue actually generate its force. We're going to talk about different types of muscle. There's skeletal muscle, obviously, and skeletal is, we're going to end up talking about fast and slow twitch. We'll talk about smooth muscle, the involuntary, which is called smooth because it doesn't look stripy. It looks just how, we'll talk about that. But basically, it's the one that's like lining your organs and your blood vessels and operating your irises and things. Has some similarities to skeletal muscle, but it also has some important differences that you do need to know. We will talk about cardiac really briefly, but we're going to spend like 90% of our time here on skeletal, like, I don't know, or probably 95% of our time on skeletal, 4% on smooth, and 1% on cardiac. So we're going to really kind of stay focused mainly on the skeletal muscle, that voluntary muscle, talk about how does it work in general, what are the different types that you get, and how are they different both physiologically and anatomically. So let's start with kind of your generic skeletal muscle cell. Skeletal, also called striated muscle, because under the microscope it looks kind of stripy. And it's a cell, because it's a long cell, we sometimes call them fibers. And it's striated because it looks kind of stripy. I'm going to say it's really long. So this can be over like 1 foot long. You have single muscle fibers, muscle cells, that extend the entire length of your biceps brachii muscle. Sartorius goes like from the top of your hip all the way down to the below your knee. Single cells, just really freaking huge cells. Embryonically, it's many cells that fuse together to create these big cells. So there's usually many nuclei, hundreds of nuclei. The cell membrane. So I talked about muscle cell contracts. The other important thing is it's excitable. So the cell membrane, I'm going to go into my typing mode here because it's going to be easier for me. So it's going to be called the sarcolemma. That's its special name. And contains voltage gated sodium and potassium channels. So what does that mean? That should be ringing some bells for you all. What else did we see that was full of voltage gated sodium and potassium channels? Oh, come on. Anybody? One person?
Touch receptors?
No. No, the voltage gated touch receptors had mechanically gated receptors.
Oh, yeah. Okay.
Yeah, those ones you stretch them and then channels open. Particularly, we had these fast sodium channels and these slower potassium channels, and they turned on when the voltage went above minus 55. Is this ringing any bells?
Action potential?
Yes, action potentials. Remember, action potentials along the axon. These are what you found on the axon that generated and sustained action potentials. So the sarcolemma here, the sarcolemma has action potentials. What is going to start an action potential to start it going in the first place? What had to happen at the axon hillock for an action potential to get started?
The threshold had to be reached.
Exactly. You had to hit that threshold. You had to be above minus 55 millivolts, and that started that whole chain reaction. In the case of the skeletal muscle cell, we've been talking about the somatic motor neurons. So here is like some axon from a somatic motor neuron. It's going to come, it's going to create this synapse. So we're going to call our neuromuscular junction here. This should start feeling familiar. This is actually just looking from another point of view, the stuff we were just talking about with the somatic motor connecting to the muscles. What does this axon release at this neuromuscular junction?
Acetylcholine.
Exactly. Releases acetylcholine. And then on the muscle cell itself, we had those nicotinic receptors. Nicotinic cholinergic receptors. So what happens when you activate a nicotinic cholinergic receptor with acetylcholine?
It opens the sodium channels.
Exactly. It opens sodium channels. It's going to, remember we said they're particularly distinctive by being always excitatory. So if you've got some action potential coming down your somatic motor neuron here, releases acetylcholine here into the neuromuscular junction cleft. The nicotinic cholinergic receptors bind that acetylcholine and sodium channels open. What happens when sodium channels open? What's going to happen to the membrane potential here?
It goes up.
It goes up, which is going to, trigger action potential, which is then going to go across the entire sarcolemma. So this is going to be an important part of what we call the excitation contraction coupling between the nervous system and the muscles. When you send a signal down that motor neuron, it's going to then cause a huge action potential that spreads out across the entire muscle cell. And we're going to see in more detail exactly what that does to actually create the contractile force. But this is the first step of activating a muscle using your nervous system, is the signal down the neuron sends a message across the neuromuscular junction, which is then going to create an action potential that spreads across the cell membrane of the entire muscle fiber. So keep that in the, put that on a shelf. That's going to be coming back when we take a look at the whole the different steps from your motor neuron activating to a muscle actually squeezing. So that's the first part then. Thinking about this big long cell that can be excitable if you send a message down the somatic motor neuron. Again, let's draw our big long cell here again. Inside there are these organelles called myofibrils. So it's going to have actin and myosin are the two main contractile proteins that are going to end up creating the contraction. But there are also other important proteins in there that we are going to talk about that are going to be critical for the control of the whole process. So we'll spend a lot of time with actin and myosin, but then we're going to introduce troponin and tropomyosin and things like that. The more myofibrils you have, the stronger overall force that the muscle cell can generate. Muscle cells that are optimized for stronger contractions are going to have wider overall diameters because they're filled with more of these myofibrils. In fact, if you've been you're working out and your muscles are getting bulkier, you don't get like bigger muscles because you're getting more muscle cells. But instead, the muscle cells you have start creating more and more myofibrils within the individual cell. So overall, you have more of these things contracting in parallel. This whole cell will be able to generate more force. And again, because it's filled with more of these, the overall diameter is going to be fatter. And that's why the overall muscle made up of these fatter cells is getting bigger looking. So myofibril. The other piece that we need to add into the overall cell is, it's called the Sarcoplasmic Reticulum. Again, this drawing is bad. I will show you a picture of this in just a few moments. Sarcoplasmic Reticulum. So obviously from its name, it sounds like endoplasmic reticulum, except it's inside your muscle cell. Things that are inside muscles often are called sarco, like sarcoplasmic reticulum, sarcolemma. Sarco just means flesh. There are little ATP powered pumps that are constantly vacuuming up calcium out of the cytosol and bringing it into the sarcoplasmic reticulum. Right, so ATP is being broken down for energy, and you use that energy to run these little calcium pumps. So the sarcoplasmic reticulum, if you think about it functionally, it's basically like a calcium reservoir. And when the muscle is just kind of waiting around, the calcium is not in the actual cytosol. Instead, it's going to all be stored in the sarcoplasmic reticulum.
That's when the muscle's at rest?
Yes, exactly. However, the thing that is going to trigger the sarcoplasmic reticulum to release the calcium is going to be that action potential across the sarcolemma that we had just talked about a few moments ago. So kind of putting this together backwards, but basically an action potential across the sarcolemma will cause calcium to be released into the cell. So that is ultimately going to be another part of this we call excitation contraction coupling. Basically, you're going to have action potential coming down our motor neuron, activating the neuromuscular junction, That then depolarizes this little part of the membrane, which is going to cause an action potential to go across the entire cell. And then as that action potential goes across the entire cell, that's going to cause calcium to be released into the cell. And the calcium is, as we're going to see in a few moments, ultimately going to cause the actin and myosin in the myofibril to pull and generate the contractile force. But again, we will see calcium's job in just a few moments. So I think at this point, we're ready to go into the myofibril and meet actin and myosin. And I actually did line up a cool little YouTube thing that I think will be pretty if I can make it work correct as well. So let's see this. So actin, also called the thin filament. Actin is made out of these little subunits, actin subunits. They connect together, but ultimately they make this long, thin thing that we are just going to call the thin filament. However, there is like this little groove. That groove is going to be important because there's another protein that actually is going to fit in there that we're going to see in just a moment. The other part is the myosin, which we call the thick filament. Myosin is a big protein. It has like 4 different subunits. Does anybody, what did we call the level of structure in a protein when you needed multiple subunits to make a functional protein? Anybody remember that?
It's a quaternary?
Yep, that quaternary, that 4th level. So you're going to have, there's 2 heavy chains, 2 light chains, create this more complicated thing. It's got a head, it's got a tail, and it has 2 conformations. It can either be what we call this kind of cocked conformation, where the head is kind of pulled back, or you can snap it forward. That's what I'm going to call like the sprung conformation. To cock it, you need ATP. So ATP would be like taking your thumb and pulling back on the hammer on an old revolver until it like snapped in place and was ready to go, right? So once it's sprung, you then need ATP to get it back into the cocked position. And then obviously we need a trigger. What is going to be the trigger that actually causes it to spring? It's going to be actually binding actin. We call it forming a cross bridge. So basically, you've got like a myosin here in its cocked position. If it is able to actually touch, woo, that is going to cause it to spring. So this is going to be at the core of muscle contraction. Myosin head here, if it actually is able to bind onto the actin, it will then grab and pull, and we'll see that that's going to actually be generating the contraction, that myosin head snapping and pulling on the actin. Obviously, if you don't want this happening, you need to have something keeping the myosin head from touching the actin. That's going to be the job of what we call tropomyosin, which sits in that little groove. So as long as tropomyosin is in the way, then the myosin won't grab and you don't actually have this grab and pull. Obviously, we need a way to control whether the tropomyosin is in the way or not, because if we want the muscle to contract, we actually want the myosin to grab. So there's going to be yet another protein, something called troponin. Let me erase this. So troponin kind of got 3 binding sites. It has a binding site on the actin, binding site on the tropomyosin. I should probably make that really look like tropomyosin and not. So it's going to be on the dag nabbit. Dag super nabbit. Here we go. So it's going to have, we can go back again. There it goes. So it has one on the tropomyosin and then another thing that binds onto calcium. So basically what's going to happen here when troponin, when the gosh darn it, troponin binds calcium, it changes shape and moves tropomyosin, which allows myosin to bind to actin. So I'm realizing as I'm typing this, especially if you're not familiar with all these words and terms, it probably sounds like just nonsense. But when you're on your own, this will all make more sense as you spend time with it. The basic idea, again, is as calcium comes in, which is coming in from the Sarcoplasmic Reticulum, calcium comes in, binds to the troponin, troponin moves the tropomyosin, which then allows the myosin to grab, which lets this thing to grab and pull. So then what we can do is take a look at how we connect this all together. You just basically have a bunch of these repeating units, are called sarcomeres. So we have actin. These kind of boundary lines are called the Z lines. Z line comes from zwischen, from German, like between. The myosins are actually connected up with hundreds of them tail to tail to make the actual thick filament. The actual thick filament is actually a whole bunch of myosins with their heads kind of facing outward. So myosin, thick filament. This is the actin in red, which is the thin filament. And when calcium comes in and troponin gets out of the way, then the myosin can grab onto the actin and it snaps and pulls, and the Z lines are going to get pulled towards each other. I think one of the important things to remember with this sliding filament theory, the actin and the myosin don't actually get any shorter. That length of the myosin, the length of the actin stays the same. It's not like a rubber band, like an elastic band or something where the actual thing actually does stretch and get shorter. Instead, you have these two things, the thick and thin filaments, and they either get deeper into the pockets or less deep in the pockets. So if you look at my thumbs are getting closer and farther, but my fingers aren't changing length at all. So in muscle contraction, it's the same thing. The actin and the myosin, slide past each other, get deeper into the pockets or less deep into the pockets. But you don't actually have a thing actually shortening. So if we go again, right, these what we call the Z lines are getting closer, so the overall muscle is getting shorter, but neither these nor these are actually getting shorter. And this is called the sliding filament theory. This dude named Huxley back in the 50s first came up with it. The idea that it's not an actual one thing getting shorter, it's actually filaments sliding past each other and the overall muscle getting shorter. I can show you, let me see if I can make this work. In the last couple of minutes here, I'm going to do my best to show you some little video.
So once the action potential goes off, that's what releases the calcium?
Yeah, the action potential releases the calcium. The calcium then binds the troponin, which moves the tropomyosin, which then allows the myosin to grab the actin and start this and make the actual contraction. So let me see if I can make this happen. I'm gonna go, I'm learning my shortcuts here. I'm gonna share screen. I think it's, where is it? It's this. All right, I'm gonna do my best here. And
this woman is using muscles to breathe, circulate blood.
Can you hear what's going on there or not?
Your cardiac and skeletal tissues are involuntary.
All right, I'm gonna get further in. So here we go.
Neuromuscular junction is the site of the signal exchange.
So here the signal's coming down.
The synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils.
So here's the myofibril.
Myofibril contains contractile units called sarcomeres.
So here's where we were just talking about.
Adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line.
Don't worry about the M line.
Of the protein actin, which are anchored to the Z lines.
Right, so there's all these things about M lines, Z lines, H bands. The only one you need to pay attention to is the Z line, which is kind of in between the sarcomeres. Don't worry at all about A bands and I bands and all that crap. I'll talk about that more later, but it's back in the day when people just knew this stuff by its optical properties. So Z line's important because those are what are getting closer when the sarcomeres shorten, but don't worry about any of the other letters that you're going to hear.
On the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through a...
So it's actually, that's an important thing to know. I don't know if you just caught that. The myosin has to bind ATP to detach and reset. So what would happen if you're dead and there's no more ATP left in your body?
Your muscles.
Right?
It will move.
Yeah, the muscle is gonna be stuck at whatever length it is when the ATP runs out. The myosin is attached to the actin. That means the muscle can't get longer or shorter. So what do we call that if all of your muscles have locked in at whatever length they're in and none of your joints can move anymore?
Rigor mortis.
That's rigor mortis. So when the ATP runs out, then the muscles are locked, and basically until like the muscle starts decaying, you're stiff as a board.
To either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When the muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the Sarcoplasmic Reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Ha 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 ha. Okay, stop sharing. All right, so we have more to say about muscles, but obviously we're done for the day. What's that?
Your own lack of talent is being interrupted by other people.
What's that? Wait, hold on. Let me, hold on. Sorry about this. I have commercials. Hold on, I got to stop it. Okay, sorry about that all. So on Wednesday, we will finish muscles, get into the cardiovascular system, and we will be doing our blood pressure lab. Remember, you have a test coming up in a week, and also I will be sending out more information about varieties of things with Cengage and things as I get it put together. But otherwise, are there any comments or questions or anything before we take our break?
So whenever our muscles are relaxed, is ATP is not binded to myosin?
When a muscle is totally relaxed, ATP, yeah, if myosin is bound, then it will they'll hold their length.
Well, okay, yeah. When is the ATP bound? Because it says myosin has to bind ATP to detach itself. So
right. So then it detaches, and then ATP is hydrolyzed. It recocks, and now it's ready to go again.
Yeah.
So it's kind of waiting. But if there is no more calcium around, then it's just waiting, and it can't grab on and form a cross bridge because the binding sites on the actin are blocked by the tropomyosin.
Okay, so is ATP attached to the myosin whenever our muscles are relaxed?
I think it actually comes in and hydrolyzes. I think it lets go actually as it lets go and it recocks. Then I think it lets go. So I think the ATP comes in, allows it to detach, recocks, and then the ATP is gone and waiting for the next power cycle. So